This video will be discussing glucose concentrations in the body. We will talk about its physiological functions when the body is at homeostasis and what pathologies might occur if the homeostasis feedback model were to be interrupted in some way. One of the main areas that we encounter glucose in the human body is within the endocrine system, so in the pancreas here. The pancreas has two major secretions. There's exocrine secretions and endocrine secretions. We will talk about what ways these secretions are involved with glucose in the body. And first, we will start with exocrine secretions in the digestive system. This interaction between the pancreas and the digestive system will occur here in the duodenum.
So I will be talking about the digestive system. So when we intake food, we intake it as a carbohydrate, which is considered a polysaccharide. And then after we ingest our food, obviously our saliva helps break it down, but in the stomach is where it really starts to get break down, and we have enzymes that break it into the disaccharides, and then another enzyme that helps break it down into its monosaccharides. So here's a picture of how it's broken down, and this is the duodenum of your small intestine. So what happens is we have our starch, and the pancreas secretes pancreatic amylase, which breaks up the carbohydrate, which is the starch or polysaccharide, and then that breaks it down into its disaccharide pieces. And after it's a disaccharide, another enzyme comes in and breaks it into the monosaccharide. So in this case, the enzyme used would be maltase, because we're going to be breaking down maltose. So after we are in the duodenum and we have our glucose, we want to start absorbing it into our extracellular fluid. So to do that, we have a sodium glucose symporter, and this brings glucose into the cell against its concentration gradient using energy that is stored in the sodium concentration gradient. So after it moves into the cell, we use a GLUT transporter, which then pumps glucose into the extracellular fluid by facilitated diffusion. The next step to this is that a sodium potassium ATPase pumps sodium out of the cell, and this is to keep the intercellular fluid sodium concentration low. So we want to make sure that we don't have too much sodium inside of our cell. In the event that we are not getting carbohydrates broken down into their individual monosaccharides into glucose, and it passes right through the system, we would be having an issue with our homeostatic feedback model not sensing our input signal, so therefore we cannot get it into the integrating center and have essentially the pancreas turn on to excrete its pancreatic amylase.
Moving back to the pancreas, we will talk about its endocrine secretions and what role they play in keeping glucose concentrations at homeostasis. This is an image of some tissue from the pancreas. You can see out here these darker colored cells are exocrine cells, so these would be the ones that were just discussed that would be secreting enzymes into the digestive system. And then in the middle here is a cluster of different cells called an islet of Langerhans. So these islets are just clusters of cells throughout the pancreas that are made up of alpha cells, beta cells, and D cells. So the majority of them are beta cells, which are cells responsible for production of insulin and amylin. And then there's alpha cells, which secrete glucagon, and D cells, which secrete somatostatin. So somatostatin is actually a hormone that regulates other endocrine cells. So we will focus on beta cells and alpha cells and how they relate to glucose. So beta cells we've seen before in this class, so here is just a review of them. This here image on your left is a beta cell at rest. So this potassium ATP channel is open and potassium is just leaking out down its electrochemical gradient. This voltage-gated calcium channel is closed, and there are low amounts of glucose entering the cell, which means little amounts of metabolism and little production of ATP. And then all the way at the end, here we have no insulin being secreted. So then if we come over here on the right, then this is an example of when beta cells would secrete insulin. So this would be triggered by depolarization of the cell. So more glucose in the blood would increase metabolism. More glucose coming in here to go through glycolysis and the citric acid cycle, producing more ATP. Then would encourage ATP to come up and bind to this potassium ATP channel gate, which would open it, close it, and then the cell would depolarize and calcium would start to flow in down its electrochemical gradient. So then this calcium would trigger release of insulin down here in step 7. So we just discussed beta cells and how they secrete insulin to help reduce blood glucose levels after eating. So now we will talk about alpha cells and how they secrete glucagon to help increase blood glucose levels during the fasting state. And this will all happen in the liver. Mostly we want to pay attention to here where this glycogen is being broken down and then gluconeogenesis is starting to happen. And then actual glucose is formed over here with the help of glucose 6-phosphate and eventually released into the bloodstream, increasing amounts of glucose. So how does that exactly happen? Well, glucagon works in many ways. So one way is here. It is encouraging the enzyme glycogen phosphorylase, which is breaking down glycogen so that it can be converted into glucose. It's also inhibiting glycogen synthase, so it's preventing this glucose from returning back into the form of glycogen. It also inhibits glucose from breaking down into pyruvates and acetyl coenzyme A, and then at the same time is encouraging acetyl coenzyme A, pyruvate, and oxaloacetate to form glucose during gluconeogenesis. Up here you can also see the hormone-sensitive lipase is helping break down some adipose tissue into glycerol and fatty acids, which then can return to be a part of gluconeogenesis again. So it's doing a lot of different things to increase amounts of glucose in the bloodstream. So again, just like we were mentioning before, glucagon and insulin are having antagonistic effects on glucose blood concentrations. So insulin is usually being secreted in a fed state when it needs to encourage more uptake of glucose and lowering blood glucose levels, and glucose is increasing blood glucose levels during the fasted state. So you can see that here where we have plasma glucagon concentration in the red, plasma glucose concentration in the dotted, and plasma insulin concentration in the green, as they relate to this meal time here. So we have, for example, glucagon concentration is pretty high, trying to increase amounts of glucose in the blood until mealtime, and then we're entering the fed state and glucagon is less necessary because there's already high levels of glucose in the blood. And then over time, as it's been longer since we ate, it will increase in amount. Down here we have basically the opposite thing happening, where insulin is very low during the fasted state, and right after a meal it shoots up because it wants us to decrease blood glucose levels and absorb some of that glucose to store as glycogen or in adipose tissue. And then the effects of that are this plasma glucose concentration right here in the dotted line remaining about steady in its normal homeostatic cycle. We've talked a lot about trading of different hormones and enzymes and amounts of glucose through different systems using the bloodstream. So what exactly is happening to glucose in the bloodstream? At some point it's going to enter the nephron in the renal system and experience filtration.
This is your nephron, and when glucose enters, it's normally reabsorbed by the time the filtrate reaches the end of the proximal tubule, and you can see that in the next slide. So by the time your glucose enters from the bloodstream, it is reabsorbed before the end of the proximal tubule because of all the carriers that are supplied on the tibial epithelium that capture the glucose. So it just is captured as it's filtered through. And then again, you can see this here. So this would be, for example, the walls of the nephron, and then the glucose is reabsorbed back into the extracellular matrix, and that usually happens through sodium-driven reabsorption, so just kind of follow sodium as sodium flows down the concentration gradient. And here you can kind of see how glucose is handled by the nephron, and usually like I said, the filtration rate of glucose from plasma to Bowman's capsule is proportional to the plasma concentration of glucose. So the filtrate glucose concentration is always equal to the plasma glucose concentration. So for the reabsorption rate of glucose in the proximal tubule against the plasma concentration of glucose, reabsorption exhibits a maximum transport rate. So that would be, for example, if there was like 8 carriers in the epithelium wall, then 8 glucoses that went by would get reabsorbed. And if there were to be more, then you would have saturation, and now all of them would be reabsorbed. So that's what the green line is here. The excretion rate, which is the blue line, of glucose in the relation to the plasma concentration of glucose. If you remember, excretion is filtration minus reabsorption. So when plasma glucose concentration are low enough that 100% of the filtered glucose is reabsorbed, then that's why the line would be straight, because all of it is reabsorbed, and then that's why the filtration line is straight, because it's all retaken. Okay, so just a reminder, glucose is not secreted, and typically with normal glucose levels, all the glucose that goes in, it goes back out, so it's reabsorbed. And when glucose levels concentrations become excessive, for example, diabetes mellitus, so type 2 diabetes, glucose is filtered faster than it is reabsorbed. So before it reaches the end of the proximal tubule, where usually all those carriers are for glucose, there's just not enough of them, so the glucose is filtered faster than it can be reabsorbed. Thus, you have reached saturation, and because there's more glucose than carriers, and so you will excrete that excess glucose since you can't reabsorb it in the proximal tubule because of the lack of carriers available because of saturation. And so what it does is just go all the way through your nephron into the collecting duct, and it will be peed out. And so this is where you have hyperglycemia, so you are peeing glucose, which you're not supposed to do.
If we were to have a pathology of diabetes mellitus, we would have an issue into the integrating center. So we would be having too much glucose come into our system, and there will not be enough channels to let all the glucose be absorbed into the renal system. So therefore, instead of the blood or the glucose being filtered back into the blood, it stays in the filtrate and it comes out and our body excretes it. Therefore, we lose glucose.
Another way that insulin homeostasis can be interrupted in the body is with type 1 diabetes. Type 1 diabetes is when there's autoimmune destruction of beta cells in the pancreas. So that would be interrupting the homeostatic feedback model around the integrating center as well, because the beta cells are not producing the insulin that they should be to control blood glucose